Hi girls, I'm going to go through all of the questions in Learning Target 1A, which is about evolution and genetic change. So to start off, number one, can you explain how a homologous structure is different from an analogous structure? So I'm going to talk about this a lot this year, and it's really important for you to start learning what these root words mean because they're going to give you clues that help you figure out problems and definitions of words that you may have never even heard of before. So I'm going to start off with homologous structures. If we look at that main root, homo, that always is going to mean the same. So we see that a lot for genetics. Homozygous is going to be two alleles that are the same. So whether that is homozygous dominant in two dominant alleles or recessive with two recessive alleles. So with homologous, we know that something is going to be the same. So that thing that's the same is their ancestry. All of the species in question are going to have the same common ancestor. From there, what they've done is they've developed some features that are going to be similar. So for example, the human arm is actually similar to a bat wing. We know that because if you were to open that up and look at actually at those bones, you'll see that they both share a humerus, a radius, an ulna, carpals, metacarpals, and phalanges. The thing that isn't the same about a homologous structure is their function. So they have the same common ancestor. Over time, they've developed some structure. But now that function might differ. As you can see, our arms function very differently than a bat wing, but they still have those similarities. An analogous structure is going to be basically the opposite. So they won't share a common ancestor. However, what they do share is the same function. So, for example, we can now look at a bat wing and a butterfly wing. Those have the same function. They help that animal fly, or insect, whatever we're looking at. It helps that creature to fly. However, they don't share a common ancestor. So homologous structure, common ancestor, possibly a different function. Analogous structure, different ancestry, common function. And I'm going to show you a picture of that now. I made a different video for numbers 2 and 4 about Hardy-Weinberg equilibrium, so make sure to watch a separate video for assistance on those questions. So I'm going to move right on to question number 3, which states, can you compare and contrast genetic drift and gene flow? While they sound similar, they are actually quite different. Before I talk about them, though, we need to know what our 4 main mechanisms of evolution are. So the first one is mutation, natural selection, which I'll talk about in question number 4, or 5, genetic drift, and gene flow. Mutation comes along a little bit later in a different learning target. So genetic drift is the change in allele frequency. So just as a review from middle school, when we have a gene, and think Punnett squares, that's like the easiest way to put it. And you're making a Punnett square, you have your 2 letters on the top of the square, 2 on the side. So the letters on the top individually are called alleles. You can have your dominant allele or your recessive allele. So that is what I'm talking about, the allele frequency, how much of those alleles are present in a population. So genetic drift is the change in that allele frequency by only random mating. So anything that is not perfect. So let's say we have 10 red bugs in a population and we have, that's my mom, sorry. It's okay, Mom. 10 yellow bugs in a population, and those bugs randomly mate. We know by Mendelian genetics that that offspring is not going to be a perfect 50-50 split. We know that because of our dominant traits, right? So we have a new generation. Let's say our new generation is of 20 new bugs, and we have 14 red bugs, and we only have 6 yellow bugs. Well, now our allele frequencies are not 50-50 like they were before, they're now uneven. So the one with more bugs, so that's the red, 14 red bugs, has a higher allele frequency than the yellow ones do. So what can you predict for our next generation? Well, based off of those uneven numbers, we're actually going to see that continue to separate because there was more in that mating pool of the red allele. So that is what happens in genetic drift. It's just that simple changing of frequencies because of random mating. Now, there are two situations in which we see genetic drift occurring more frequently. You'll talk about these a little bit more later on, but I'll introduce you to them now. The first one is the bottleneck effect. So bottleneck is when there is some sort of a natural disaster, for example, that greatly reduces our population. So we start with a population this size, we have a natural disaster, so that's going to reduce our population. So we now have, you can go through, Kat. I'm visiting my family for the weekend, so here's my sister too. So we have a large population. Natural disaster is going to reduce our population size. So now we only have this much. Well, what happens when we have a smaller population is that we have actually a smaller gene pool. We have a smaller gene pool, there's less alleles to go around. So let's say the ones that survived were this time our yellow bugs, mostly yellow bugs. Well, once that population starts to expand again, because that gene pool was mostly the yellow bugs, well, now our allele frequency once again is going to shift because there was more yellow bugs mating. The second situation that we need to talk about is called the founder effect. The founder effect is exactly what it sounds like. You have a small group of individuals, they're going to go somewhere new and start a new population. Well, the individuals that were founding that population are the gene pool. So if you have mostly yellow bugs, well, now your gene pool is mostly yellow. So the offspring are going to probably favor that original color. So bottleneck effect and founder effect are really going to influence genetic drift. Now we also have something called gene flow. Gene flow is when you're going to have individuals or groups of a species that are going to migrate to a new population that already exists. Now once that population that already exists in this new group enter, well, you're introducing more traits. So that's going to be more genetic information that's going to be passed along to the new population. So that's going to be gene flow. Genetic drift is changing in the allele frequency. Gene flow, new traits or new genetic information is flowing into a current population. So that is different than the founder effect because in gene flow it actually has to be entering a current existing population. All right, I'm going to move on to the last question real quick, which is, can you explain how natural selection acts as a mechanism for evolution? So, right here. With natural selection, what's going to happen is that certain alleles are going to become more or less common over generation. Natural selection is basically when you have an advantageous feature, then nature is going to decide which ones are going to be able to survive, and the stronger, more advantageous features are going to be the ones that are passed along. The most common story for natural selection is from the peppered moths and the Industrial Revolution. So before the Industrial Revolution, there was a dark peppered moth, which was black-bodied, and it was pretty rare. So in our allele frequencies, that one would be much lower, and this was in the UK. And the other peppered moth actually was white-bodied. And then what happened during the Industrial Revolution was that the trees in which the moths lived on became covered with soot from all these, oh my gosh, what? Factories, thank you. I couldn't remember the word. From all these factories being built. So all the trees start becoming covered with soot. Well, that soot is more going to be that gray-black color. So now all of the white-bodied moths were easily spotted by whatever was hunting them. So usually birds and bats. So they were going to be getting eaten much easier. Whereas the black-bodied peppered moth blend in to the trees now. So we actually saw a huge shift in allele frequency because of natural selection. Nature decided what was a more advantageous feature. At first it was the white-bodied because it blended in with the tree. Then because of the Industrial Revolution we have this switch, and now all of a sudden the black body becomes more advantageous. So that's our main reason for natural selection, acting as a mechanism for evolution.